Welcome to Lab 3, where we'll watch tiny transport rules create real, measurable changes.
Today we're exploring cellular transport mechanisms, simple diffusion, osmosis, and filtration.
First, the cell membrane is selectively permeable. This means that not everything is able to cross the membrane.
Movement through the cell membrane depends on factors like particle size, charge, and the concentration gradient of the solute. Diffusion is the movement of a solute from an area of high concentration to an area of low concentration. The rate of diffusion depends on many factors, such as the concentration gradient of solute, the temperature, and the size of the particles.
To investigate the principles of simple diffusion in lab this week, we will perform two experiments. One will look at how particle size affects the rate of diffusion using agar plates. The second will use dialysis tubing to investigate how particle size affects its ability to move through the pores of the tubing.
Now for osmosis, the diffusion of water through a membrane. During osmosis, water moves from an area of high concentration to low concentration. This means that water will move toward the area with the higher concentration of solutes.
To investigate osmosis, we will perform two different activities in lab. The first will be to determine the rate of water movement from an area of high concentration to low concentration using an animal membrane bag. The second will be to investigate how water moves when a de-shelled egg is placed in isotonic, hypertonic, or hypotonic solutions.
The last cellular transport mechanism that we will investigate is filtration. Filtration is pressure-driven. In this case, both solutes and water will move through a membrane from an area of high pressure to low pressure. In the body, the pressure is created by blood moving through blood vessels. We will recreate this in lab using gravity and the fluid volume to create a pressure gradient.
During lab, a mixture of charcoal, starch, and copper sulfate will be added to a funnel lined with a piece of filter paper. You will count the filtration rate when the funnel is full of liquid and when the funnel is half full of liquid. Additionally, you will then test the filtrate produced to see what was able to pass through the filter paper.
In this introductory video, we learned about simple diffusion, osmosis, and filtration. To summarize, first, simple diffusion is the movement of a solute from an area of high concentration to low concentration. Second, osmosis is the movement of water from an area of high concentration to low concentration. Lastly, filtration is the movement of solute and solvent from an area of high pressure to low pressure.
To finish preparing for lab, be sure to read through the lab in your manual. Once you have read through everything, take the short pre-quiz to assess your preparedness for lab. Remember, the pre-quiz must be completed prior to the start of your assigned lab. See you in lab.